All right. Okay, thank you. Thank you for the thorough introduction. As Gonzalo said, I'll be talking about some of the work in my lab that's kind of inspired by general problems with trying to infer science from data. And can we really get systems like deep learning or machine learning broadly to actually, discover anything scientifically? So we've generally seen that to some extent, deep neural networks can automate things like stuff pathologists do, for example. But that's something pathologists can already do. Can we do something that pathologists can't do? Can we discover brand new science? So that's been kind of the focus of my lab. And that's in a sense why, because the question is so broad, that we've been dealing with a lot of different kinds of data in our lab. So the majority of data, whether it's single cell RNA sequencing, molecular data, which I'll talk a little bit about today, which is molecular structure and function data, as well as fMRI data, which is a type of signal on the brain that's measured at some level of resolution, changes with time. What these all kind of have in common is they're basically very high throughput, high dimensional, noisy data types. And they're often generated on MOS these days. There's even atlases, like a cell atlas. There's the Seattle Brain Atlas that generates these kinds of data sets. But then the question is, what are the insights that we're really going to get from these? And can we use machine learning to basically detect and extract patterns and things like that? The things that I want to highlight about these data types is, first off, if you just tried to look at them with very generic techniques, for example, something like PCA, it's going to be very, very noisy, so noisy that it'll probably drown out the signal. They're also very, very high dimensional, but the dimensions are redundant a lot of times. So they carry information within them, and they have a lot of mutual information between the variables. So that's where some of the lower dimensional manifold learning stuff that I'll talk about comes into place. And the final thing is most of it's unlabeled because, for example, cellular data. People might tell you that there's 10 types of cells, 20 types of cells. Not really, because each cell is its own individual, so to speak. So actually there's trillions of kinds of cells, right? So it's all unsupervised. So how do we make machine learning and deep learning techniques really, up to the standard of discovering something from this kind of data? And so I would say basically you need to sort of increase the power of the machine learning techniques. So this might sound kind of surprising to you because, okay, we have Chat GPT that can, I don't know, beat the Turing test or talk to us or something like that. But actually, when it comes down to meeting biophysical constraints and actually discovering biology, we usually find that they lack enough expressivity and power. So some of the techniques I'll talk about is actually trying to increase the power. Today, particularly, I'll be talking about increasing the power of graph neural networks. The second thing is, if you're generating, let's say in biology, some kind of cellular measurement after perturbation or something, it has to meet biophysical, biochemical type constraints. So deep neural networks especially aren't super good with constraints. You sort of have to bake them in. There's a few different ways you can do it. You can do it by regularizing. That's not actually constraining. That's like kind of nudging. Like, oh, don't do that, or I'll penalize you or something like that, right? It's like nudging a kid or something. But you could do it in a more heavy-handed fashion if you bake it into the architecture. So that's one of the things that we do, although I don't know if I'll get a chance to talk extreme amount about it. And the final thing is we want these techniques to be interpretable. It's not enough for them to make predictions. We need these to be interpretable. And so if you take all these kinds of things together, what you'll actually have is a sort of expressive system that, the long-term vision would be to have an expressive system that at some level of granularity embodies the actual biophysical, biochemical constraints that you can interpret. So it's almost like you have something in between a prediction and a simulation. So that's what I would kind of claim. And so these kinds of systems are still sort of coming along. Scientists are still discovering them. I was actually at a very beautiful retreat just a week ago in the south of France, where a bunch of people that are using deep learning in, physics, chemistry, biology came together and to talk about it. And these were basically what they were talking about. So today I'll be giving examples on neuronal systems and molecular systems, more as a way to kind of motivate, but I'll also present some of the results. There's messages on chat. Is someone checking those? Okay. Some of the things that we are interested in doing that may be a little bit different than the static prediction is, we would also be interested in predicting and characterizing dynamics of these systems, because most biological entities are dynamic, and also sort of comparing the dynamics. So I claim that for a lot of these, a really good starting point to give an interpretable model on which you can, conduct dynamics, that graphs are a good substrate. And I don't think I really have to convince anyone in the audience that graphs are really important, because you're all here. But basically, the things I like about graphs is that they can capture many different kinds of structure, from just interconnection structure to underlying geometric and topological structure. And they're very flexible. They can model almost any kind of biomedical entity, from molecular networks to interaction networks to just, similarity diagrams. And they, in essence, enable a lot of other kinds of math and constraints to be put in. So this is kind of the theme or pattern that we've been following to increase the power of basically deep neural networks in this talk, but also other methods, is we try to combine graph signal processing with geometric and topological information, and we try to imbue deep learning networks with those so that they're still learning, but they have these kind of fairly generic priors built in so that they can do these other things. So if you want kind of an example of a successful system in deep learning that has already done that, it's CNNs. CNNs are sort of the poster child, or convolutional neural networks are sort of the poster child of success in deep neural networks. And you can see that they have some of these things kind of built in. So they're usually made for naturalistic images. So in medical images, they function differently, but for naturalistic images, they have these convolution operations built in that enforce translational invariance. They have multiple layers, which can enforce some amount of scale invariance. And they have very flexible filter functions that can pick out different features. So one of the questions was, can we extend this type of thing to graphs? This is kind of the question that motivated, for example, graph neural networks. So graph neural networks generally, and I'll cover that in a slide or two, have fairly simple operations that they regard as convolutions. So one of the things we became interested in looking at, because even early on, we were looking at these more complex biochemical entities like molecules, is seeing if there is something that could enhance their power. And we happened upon scattering, which was being developed at this time for graphs, for the setting of graphs. So scattering, some of you may know, was developed to understand convolutional neural networks way back in 2012. And then some people, as actually some people told me yesterday, they're not really needed for convolutional neural networks because convolutional neural networks are so powerful. But the thing is, graph neural networks really aren't. So we started looking at incorporating geometric scattering into kind of graph-based learning frameworks pretty soon after they were extended. So in 2018, 2019 timeframe is when they were coming out. So one of the things we wanted to do is we wanted to keep the structural or architectural constraints of something like geometric scattering, but put it within a learning framework. So here we introduced something called learnable geometric scattering. The first version was published in MLSP, and after many years, we just got it out in Transactions of Signal Processing in 2024. This idea that you can have a learnable framework for geometric scattering. So just to give you kind of an intuition for why this might work, you might think of a molecule encoded as a graph. You can have either the bonds or the actually 3D affinity encoded in the form of a graph. And you might be thinking of sort of the field of geometric deep learning. So if you took this kind of molecule and you put some kind of signals on it, whether it's natural signals or artificially generated signals, and you put it through a graph neural network. The basic message passing operation gathers information from local bonds and aggregates them into a vertex. So basically what it's doing is smoothing or local averaging. So if you do this just once or something like that, you'll get very local features that might not span the molecule. If you do it over and over again, all the vertices will have the same signal. So you're kind of stuck between a rock and a hard place. So this is some of the problem, and this is what we're hoping that scattering can help these problems of over smoothing and underreaching. So now let's look at a little bit of the wavelets that are used in scattering. So one of the kinds of wavelets we use, and it's not the only kind that's available, is diffusion wavelets. So if you think about local feature aggregation, let's say you have some feature on this graph, we like to think of features as signals, then these two graphs, which could relate to molecules, almost have the same embedding of every node, and graph neural networks tend to concatenate node embeddings to create a graph embedding. So they'll have almost the same embedding. But in terms of molecules, these could be very different because this has this ring and this doesn't have a ring. And I think you guys have heard rings make molecules very different in general. Now let's look at a different kind of operation. So diffusion. So one scale of diffusion is actually pretty similar to the message passing graph neural networks do. So that's just created by a diffusion operator. So you can take the graph adjacency matrix, row normalize it into diffusion probability. So this creates a transition operator that can perform, discrete diffusion dynamics. And so if you start looking at different scales of diffusion, so one time point of diffusion of this signal that's at vertex 1 will diffuse the signal to its neighbors, and the next scale will diffuse it to the neighbors of the neighbors. And already you see that the different time scales of diffusion act differently on the first graph than the second graph. So this is the setup behind sort of the intuition behind why graph wavelets are useful. So these are these diffusion wavelet constructs, which were actually invented way back by my colleague, Rafi Koifman, back in 2006, way before sort of these kinds of networks became popular. And this is sort of a bank of wavelets that's created by subtracting different scales of diffusion. And P is the same diffusion operator that I was just showing you. If you want to think about it a little bit spectrally, what the diffusion operator actually does, it does sort of a low-pass filtering. So if you raise the diffusion operator to different time steps or T, in the spectral space of the graph, you get different amounts of low-pass filtering. And actually, we've used that alone to denoise data that can be modeled as being on a graph. But diffusion wavelets are actually more powerful. Rather than just smoothing, you can make them attuned to different frequencies and shapes of signals on the graph because you can co-locate them in different places. So these kinds of wavelets are the workhorse of geometric scattering. So what we do is we create a cascade of diffusion wavelets that creates sort of a deep wavelet transform. And generally it's interleaved with nonlinearities. And here I'm showing the modulus nonlinearity, but later I'll come back and show you how you can actually use a nonlinearity from deep learning ReLUs to make scattering even more expressive. So then once you have these, you can actually even put uninformative signals on the graph, take their deep wavelet transforms, so to speak. And then you can get whole graph level representations by aggregating these signals. And the aggregation doesn't have to be sums, it can be statistical moments, or it could even be a complete MLP. So now how do we make this learnable? The way we make this learnable is we build this kind of structure into a recurrent neural network, but we leave things for the neural network to actually learn to tune itself to the data. So one of the things that's learned is the different scales of diffusion. Which scale should we use? Another thing that can be learned is the aggregation, which could be an MLP. And so these are embodied in this fixed versus learnable geometric scattering. So just to go a little bit deeper into this architecture, so diffusing amounts to reapplying this diffusion operator over and over again. So this is a recurrent network that applies this operation over and over again to the graph. So to select the scales of diffusion, you can actually have this selection matrix. And this selection matrix can be implemented to roughly enforce one hotness, so one scale per row, using a softmax activation in a deep neural network. So once you have this selection, then what you do is you gather the selected scales, and they can be, increasing in their number of steps, and you subtract them off, which creates your wavelets that you're actually going to use, and these wavelets will be tuned to your data. And once you have these wavelets, then you can gather them using the scattering and aggregation module. And then you can repeat this entire construct over again. If you repeat this entire construct, then you get a deep neural network. And it's not suffering from the problem of oversmoothing, basically. There were some questions about if these kinds of more flexible geometric scattering networks would have some of the desirable properties. And it turns out that we can relax the dyadic scales to any increasing integer scales and have this dual bounded structure on the wavelets that are constructed from it. So the reason these dual bounds are important is the lower bound basically says we won't squash signals together, so they'll be distinguishable. The upper bound basically says that you won't expand noise in the signal, so the noise won't drown it out. And as I already mentioned, the intrinsic operation in the diffusion already low-pass filters noise out to some extent. If the data is more noisy, you'll see higher scales emerge from the learning. If the data is less noisy, then you'll see more high-frequency scales that naturally emerge. And of course, it still retains the permutation, invariance, and equivariance. So at the node level, it's permutation, equivariant. At the graph level, because of the scattering, because of the aggregations, it maintains permutation invariance. So we can put these kind of scattering layers within larger neural networks that can become end-to-end differentiable. So we've shown that we can do this in kind of an autoencoder framework for different applications that I'll show you. We can also put it in within a generative modeling framework to generate molecules. But I'll also be talking about a task that people in GNN literature don't talk much about. So autoencoding can be generating an entire graph, representing an entire graph or a graph representation. Generative modeling can be generating the entire graph. But there's another task that's actually very important in the biomedical sciences, and that's signal level tasks. So that's not node tasks, it's not edge tasks, it's not a graph task, but it's a signal task. So what does that mean? A signal is a pattern over the whole graph. And there's a need to classify signals or regress signals to a value. And I'll show you that. To give you an example that I'm not going to go over, if you have a cell-cell affinity graph, then genes are signals over the cell graph, how much of a gene the cell expresses. And the pattern of gene expression over the manifold can say something about the gene. Is this a housekeeping gene? It's very diffuse. Is this a very localized gene? That means it's indicative of a particular phenotype, et cetera. So how do you put geometric scattering within a larger neural network? You can create an architecture from them. For example, one of the first architectures we created was a geometric scattering autoencoder, where here we have the learnable scattering transform. And then over here is the autoencoder path. And furthermore, the latent space is usually dual regularized in our applications to regress some kind of property that's important. But one caveat here that we always use deep learning to get around is that the scattering transform is not easily invertible. And I'll come to that in a bit. So we usually use either a decoder or a generator to generate from it. One of our key applications, especially initially, was biomolecular representation learning. So the idea here is you have sometimes thousands, hundreds of thousands of molecules, and even these days, even millions of molecules. And you want to represent them in a way where you can sort of reason about them, where first off, you want dual organization by structure. So points that are nearby have similar structures, at least as represented by the graph and its features, some kind of property, as well as you would want the embedding to be invertible or pseudo-invertible so you can generate from it. So already we were able to see that when we are embedding in a paper that came out several years ago, non-coding RNA, which have structures, that the geometric scattering autoencoder is pretty faithful to the known biological system. So here you have a toy graph trajectory. So what's happening here is there's a single graph, and along this trajectory, we're mutating sort of one edge at a time randomly. So this is a trajectory of graphs. So we see that the geometric scattering autoencoder shows a neat trajectory of graphs, and a lot of other things like regular graph autoencoders, the WL kernel, and these kinds of things don't really show that structure quite as clearly, or at least they don't denoise the structure quite as well. It shows up even more in the biomedical context. This is a RNA molecule that is called a riboswitch. In one configuration, it can inhibit the transcription of another gene. In another configuration, it's inactive. So it actually has two low energy states. And if you look at these embeddings, then this is very simple. This is like a 0th level sanity check. Do you see two low energy structures? And in most of the graph learning methods, you don't. So they're not capturing something fairly simple and fundamental about these molecular properties. More recently, we've become interested in using this scattering module to also kind of mimic molecular dynamics. So as I was telling you, there's things like AlphaFold that can predict one structure, but a lot of times these molecules keep changing. So they're undergoing some kind of dynamic, and we're very interested in putting forth the dynamics and sometimes interpolating them because they're very expensive to actually conduct a full simulation of. So this is a manuscript that's on archive. It's under review. So the basic idea is we encode proteins as graphs and the residue identities as signals on the graphs. We use the learnable geometric scattering. And important for this application to be interpretable, we use two kinds of attention. So this is different than the attention you see in graph neural networks, which is basically attention over nodes. Here, we can also have attention over scattering coefficients. So we can do dual attention, which is over the nodes and over the scattering coefficients. And then we put it into an autoencoder, which is regularized using our manifold learning method FATE to give you a geometric representation. And interestingly, it's regressed against time as well as the 3D coordinates. So you can decode from it into the coordinate space, and that the embedding is organized by time. So when you actually move in the latent space, you see something like a trajectory like this that's folding. And this is actually done on one of the RNA molecules, not the protein molecules, just for simplicity, because proteins would be hard to visualize here. So when we put several proteins through these, what we're actually doing is we have a coarse-grained simulation data, and this data was actually released by DE Shaw, and they have a coarse-grained simulation for several proteins. Then here is the embedding that we get from Protescape, our network that incorporates scattering. So you see that the trajectory is going something like this. And interestingly, what you see are attention paid at different points in time. And when we look at that, we see that the attention is focused on different residues, and it tends to be the most flexible residues that it pays attention to over points in time. And then you can read out what this flexible residue is, which is mainly creating the dynamics. So you see different conformations. You can take this dynamic and you can split it into different conformational regions. And then you can use the residue level attention as well as the scattering level attention to interpret what's moving and what's happening in these dynamics. So you can also segment these dynamic regimes. The way we segment is using a curvature-based segmentation of the trajectory. And so in this case of this molecule, when we segment the trajectories, we get two separate trajectories. This squiggly 8 type thing is going here, and this circular thing is here. And it turns out these correspond to two conformation that the molecule switches between an open loop and a closed loop conformation. Okay, so now just shifting applications briefly to molecular generation. In order to perform generation of the entire molecule, it's a little bit harder. So we trained an entire generation path. First, we trained a GAN, and in the recent version, we actually trained a diffusion-based generative module that comes out of this latent space. Again, because scattering itself is a little bit hard to invert. So here we have an adjacency matrix that we're generating, but the adjacency matrix then is put through more layers. For example, we have a discriminator that kind of helps rectify the adjacency matrix to be actually doing post hoc correction. We've compared the ability of this to generate, and it functions on par, if not better, than most of the molecular generations out there, but with basically a lot fewer parameters, because a lot of the parameters are reduced by using scattering. Currently, we are actually collaborating with a group in our university in ophthalmology. So this is actually unpublished. What you see here is embedding of all the drugs on a database called Drug Bank. So you can download the drugs from Drug Bank. And what we then did is, and it might be a little bit hard to see, but the red dots are some molecules that they had found from panning a molecular library against some retinal epithelial cells. And these are epithelial cells in your eye. These are ophthalmologists, and they're looking to cure a disease called age-related macular degeneration. And some of their hits are there, but their hits sometimes don't have, the idea is you take some tissue from a dead person usually. We do have a rapid autopsy program at Yale, and then you pan these molecules against them. And in culture, there are some hits that work, but they may not work in an actual person because they have too high of toxicity, or they can't get through some kind of barrier. And so what we actually do is we take this latent space and we actually move it in the direction where there's better property predictions. And that's why we tend to have property prediction as a side goal from our autoencoder structures. So what you can do is if you have two hits here, you can interpolate between them and see how the properties are changing. And you can actually do what's called gradient ascent of the property. So, gradient descent is what you use to train neural networks. Gradient ascent you can use in the latent space to improve the properties. So you can directly go in directions that are more promising and generate more promising molecules. One other thing that we were able to do is sometimes when you kind of pan a library of molecules against something, you actually don't know what they're doing. Even some of the drugs we're taking, we don't really know what the mechanism of action is, for example. And you can help uncover that too. If you have some hits here, these were some of their other hits. They had something like 5 hits from a library of like thousands and thousands of molecules. You can look to see what is the nearest neighbor in Drug Bank, what are its mechanism of action. That's not going to be necessarily the mechanism of action of the present molecule, but that generates hypotheses. So I do want to emphasize that a lot of times what we're doing is not proving these things work so much as generating very promising hypotheses. So imagine you had to test all the drugs in this embedding. That would be very expensive. But if you have to test a couple of them where we say, okay, we think this is the mechanism of action, then they can think up another assay and they can do it feasibly. So now switching from these graph level tasks, which is basically what I was showing you, capturing graph structure and graph properties. Now I'm going to move to signal level tasks. So very recently we had a publication in AI Stats that Charles, who's here somewhere, was also involved in. And here we created a slightly more enhanced version of the learnable geometric scattering because we started looking in addition to graph level tasks at these signal level tasks. So let me tell you what I mean by signal level tasks. So if you have a graph, a signal is a function that's defined on all the vertices of the graph. And the signal level task is to come up with a label for the entire signal based on its pattern of representation. So here, let me show you why this, if I can figure out how this, oh, it worked. So this is actual fMRI data, and you see it changes over time. Each time snapshot you can think of is a different signal that is appearing over the same graph. And what do we want to do with these signals? There can be many kinds of signal tasks, but one thing that's often done in fMRI is to figure out, what the internal state of the person is. So is there a particular emotion label that they have? And there's some training data to this because people write down what emotions they've had when doing different tasks or different movies. So if they're watching, are they indoor or outdoor? Are they resting or active? Or even reconstructing features of the simulation. And you can see these are all signal level tasks because the same graph, just the signal is changing over time. So if we want, scattering, it's already pretty expressive in a lot of ways. With respect to signals, then, the desirable features are something like if two signals are distinct, even a little bit distinct, we want their representations to be distinct. We don't want to dissipate energy in different parts of the signal because we don't know where the information is. And it would be interesting if we could recover the signals in the sense that in kind of an auto-encoding sense, meaning we're capturing a lot of information about it. One of the things that we noticed is for signal level tasks that the modulus nonlinearity in scattering is actually affecting its expressivity, although it doesn't seem to affect the graph level tasks quite so much. So the basic idea is that because of these absolute values, it can't distinguish between certain specific signal patterns, for example, this one and this one, because it's turning these all sort of positive. And this is not an isolated example. You can have more complicated examples. For example, if the graph is bipartite, then you have indicator vectors. And if you use these kinds of indicator vectors and different linear combinations of them, you might not be able to distinguish. So instead, what we sought to do was replace the modulus nonlinearity in scattering with ReLU, which is a very commonly used activation for neural networks anyways. But instead of using one ReLU, we use two ReLUs. So we use ReLU of the signal and ReLU of negative of the signal. So we're retaining information in negative and positive senses of the signal. And the other change we make is at every layer of the scattering, we're using the entire wavelet filter bank, including a low-pass filter. So instead of the network having to be residual, like our recurrent networks are, they output information at every different layer. So first off, we showed that if we put these in as the nonlinearities, then you get this bi-Lipschitz property. And again, this bi-Lipschitz property turns out to be useful because, again, you don't squash quite as much information in the signal. Secondly, I mean, this is sort of an example, but theoretically speaking, you can recover these signals because it's invertible. This is an injective transform in the case of diffusion wavelets. Here, for example, is the first activation minus negative of the second activation, and you can use that to recover properties. So we use this on the fMRI data that I was talking about. So this is also publicly available data. It's people watching sort of a cartoony movie. It's called Partly Cloudy. You can watch it somewhere. The idea is to classify the emotion that the person is undergoing when they're watching, different parts of this movie. And here we have sort of a, cast of different graph neural networks that perform pretty badly on these because actually graph neural networks, people haven't thought about signal level tests too much. Here's the regular scattering, but we see the BlissNet is actually much more accurate on these fMRI related tasks. Now that I've shown you a little bit about how, we've enhanced scattering, incorporated into learning framework, I'm going to talk about an even larger framework we've incorporated scattering into by also using topology or topological data analysis to be more precise. So this now goes from looking at single time points as a signal to looking at the dynamics of some signal, just like the fMRI data that I was showing you. And here we're introducing a technique called graph scattering homology trajectory. We initially developed it and published it for looking at signals from cells. So these are calcium signals from cells, but calcium signals are also usually measured as well as fMRI on brain states. And now we're extending this to also looking at neuronal data from these domains. With the initial paper on the cells, you can already see in this journal of cell biology. So now that I've given you all this sort of background on, scattering and and learnability and all this stuff, so I can tell you that basically we have these signals that are measured on different voxels of the brain, or sometimes other units. There are even recordings that are at the neuronal unit. It doesn't super matter for us. We usually put these on initially a pretty uninformative graph, and if I have time at the end, I'll briefly touch upon how we can even learn the graph, because that's the flexibility of a learnable framework. You can learn all the way till the end, back down to the graph. So we place these activation signals as signals on these graphs. We perform legs, more recently using the Bliss scattering transform. And then we get embeddings of individual time points, but then we chain them together using a couple of other techniques. This I'll skip because it's too neuronal. So what do we chain together? We chain together an encoder that's regularized with our FATE method to create a trajectory of these signals as they're appearing in the brain. So what does that look like? It looks a little bit like this. So the main thing you need to do to achieve this is to say the embeddings of two different time points, if they're creating a similar pattern in the brain as measured by our manifold learning technique called FATE, or if they're very close in time, that they should be embedded similarly. So why close in time? It turns out this is actually really important, but maybe you'll see it in a minute or two. Okay, first let me explain to you what our manifold learning method is for regularizing the latent space. This comes from our FATE algorithm, which was published a few years ago. And we think of FATE as sort of a more geometric alternative to something like T-SNE or UMAP or something like this. So what do I mean by a geometric alternative? It's geometric in the sense that it keeps a certain sort of distance, which those methods don't because they're neighbor embeddings. And it's okay because, basically if you're going from very high dimensions to low dimensions, you have to pick what information am I going to preserve? You're not going to preserve everything, right? So the way this method kind of works is you can have data in high dimensions, or in our case, we have these scattering coefficients, which are quite high dimensional because of all the scales of wavelets we choose. And then you measure distances between these. And then you turn the distances into affinities. And actually, then we turn these affinities using our old friend, the diffusion operator, into these random walk matrices, and we power them. But now we realize that rows of this matrix are actually probability distributions. And we actually take an information theoretic divergence between these rows. And this works to give us a very robust manifold distance in the case of high dimensional, high noise data. And we just embed these new distances with metric MDS. So this is the FATE algorithm. But the FATE algorithm is not good enough for these neuronal trajectories, even though it was good enough for cells. The reason is because these have even more noise. So what we're actually doing is we're actually putting two kernels into FATE. One is the kernel that I just described, which you can think of as kind of a similarity kernel, similarity between time points. And another one is a temporal kernel that enforces basically autocorrelation in the signal. And that's really what gives us these kind of trajectories from our scattering coefficients. So if you're wondering if this is really necessary, you can take a look. As you increase the noise in these neuronal type features, PCA collapses the trajectory almost immediately. UMAP and even FATE does do it a little bit later. And then T-FATE is the one that kind of retains the structure the most. So this is published in Nature Machine Intelligence last year, so you can take a look at it. So once we have these trajectories, we actually just quantify them using topological descriptors. The most basic topological descriptor you can use is these Betty numbers. So these are what are used in persistence diagrams. So if you're not completely familiar with it, I'm just going to go over a brief overview. The idea is in topological data analysis is you have some object that's hard to get your hands on quantitatively speaking. It's either sort of a cloud, or in our case, it's some kind of squiggly trajectory. But what you actually want for machine learning to work is just a set of numbers that describes or quantifies it well. So one thing that turns out to be helpful is to measure some fundamental thing that is especially easy to do, for example, on a graph, is the number of holes in the data, the number of connected components, and these sorts of things. So the number of connected components is Betty 0. The number of cycles is Betty 1. The number of cavities is Betty 2. And you can think of, higher dimensional holes. So, in, pure topology, you've seen that these have nothing to do with sort of geometry, but in topological data analysis, they're completely geometrically determined. If usually, as you grow an epsilon ball, you sort of create a graph, and then you count the number of components in the graph. And you can keep doing this as you grow these epsilon balls, and then eventually you can, maybe get a cycle. And what you're recording on these persistence diagram is which iteration this feature was born in and which iteration it died in. And you're usually interested in off-diagonal features. So just to keep you going along, because this is quite a complex thing. So we had scattering. We captured each time point using the scattering. Then we chained the scattering together geometrically into a trajectory using fate. And now we're quantifying that using homology. So why should we do all this, right? So this is actually the actual problem that some cell biologists at Yale actually came to us with. I'm hoping that this animation also works. Okay, yes. So they discovered that epithelial cells in like hair-like niches were flashing calcium like this. And they don't exactly understand why they're flashing calcium because the function of these flashes isn't known. They had questions like, are these flashes random? Are they just flashing, just a flash? Is there any kind of coordination underlying them, even a little bit? And when you capture it with our pipeline, scattering, fate, and topology, what you get are these flashes are not at all random. So here's an example of this. This is the flashing you're seeing from the epidermal basal layer where your skin follicle niches are. And actually the trajectory that's formed is super smooth. And if you compare that to the same kind of calcium flashes that are happening in your cortex, visual cortex, for example, put it through the same technique, GSDH, you see that these flashes seem more random, probably because there's more response to stimuli. So these flashes are actually very coordinated and very, very smooth. So then the biologists started looking for what is creating this coordination and what is actually happening. So it turns out that these flashes are essentially some kind of homeostatic signaling. So the cell is panning out these flashes and saying basically, is everyone okay? Is everyone okay? And if cells aren't okay, then this smooth flashing is disrupted. And one way you can make cells not okay is if you freeze them in a particular cell cycle. So it turns out cell biologists are able to do this. And if you stall them in a cell cycle, they're no longer so smooth. Or if you take other parts of the cell cycle, other parts of this mechanism, for example, something that is responsible for producing the calcium, then you'll see the same kind of effect. Then why do we need the topology? That's because this is kind of a visual description and we want a quantitative method for quantifying it. So what we actually did was we took a whole bunch of persistence diagrams of these calcium signals under various experimental and perturbation conditions. And then we took their persistence diagrams, and it turns out you can measure optimal transport or Wasserstein distance very reliably between these persistence diagrams. And then we use that, and then we're able to cluster these into different kinds of regimes, dynamically speaking. So this is kind of a quantification and clustering of the entire dynamic rather than one particular kind of signals. And then we found other kinds of players that are very important for this. So, what is it about stalling at G1 that disrupted the dynamics? You can find molecular players that I sort of go into. You can also see that the long range coordination has certain molecules strictly controlling it. So if you actually perturb certain molecules, you don't get the long range coordination. So you get this trajectory sort of breaking. More recently, we've been interested in using this again in a learning framework with neural signals. So one of the things we've noticed is that excitatory and inhibitory neurons together can create oscillations. So as a test, we used the Kuramoto mathematical model of oscillations, and we actually used these topological descriptors that we get to actually predict the parameters that are involved in this. So you can again see it in the trajectories that you see that different kinds of quantifications and stalls appear differently in the trajectory, but we can actually recover the simulation parameters with more accuracy and low mean squared error. We can classify brain dynamics, and actually we can also classify the visual stimuli that people are looking when they get these dynamics. This application, I might sort of skip. This has to do with theta and alpha waves and how we can actually predict what kinds of clusters of oscillations are in the brain dynamics. And then probably the last topic I want to cover is, after sort of going through all this, we're actually, taking a step back and saying, okay, why is it exactly that when we incorporate, these wavelets and this kind of scattering setup that things are being improved? Let's, kind of get to a little bit to the foundations of this. So we're starting to think about, a hypothesis that it's actually that we have another kind of dynamic going on, that these are dynamics of signals on graphs underlying these scattering and wavelet transforms. So when you look at message passing itself is a type of dynamic. And I think Michael Bronstein and other people have done a great job showing that this dynamic is actually a diffusion dynamic on the graph. It's a very simple diffusion dynamic. But what we're doing with scattering is even taking this simple diffusion dynamic and looking at different time snapshots of the dynamic. And already that improves things a little bit. So can we generalize this? Can we use other dynamics? And can we use snapshots of these other dynamics? So this is what we're trying now. So we're looking at continuous time dynamics now, rather than the discrete Markovian dynamics of the diffusion operator. So this is continuous heat dynamics, which is sort of a continuous version of the diffusion dynamic. These are wave dynamics. This is created by a wave equation and different time snapshots of that. And we're also trying this dynamic, which depending on its parameters can be either in chaos or at the edge of chaos. So what you can do from these different dynamics that you run on a graph is you can take signals, spread them according to different dynamics, and take different time snapshots of them, and then basically use linear combinations of them mimicking the wavelets that we had previously created. It turns out that we're seeing in our preliminary results that these are very interesting because they're correlated with topological properties. So for example, you can look at cycles and cycle lengths, and intuitively you can see that if you used waves of different frequencies, you can recover cycle lengths, or if there's a cycle. It actually turns out that our network, which uses these dynamics, which is called DIMAG, actually is very good at predicting sort of the entire extended persistence homology of the underlying graph, more so than a lot of other neural networks. We can also show that even the heat dynamic can recover another property which is more geometric, which is curvature. So you can think of curvature on the graph if you use the framework of the Olivier-Ricci setup. It turns out the Olivier-Ricci setup is basically looking at the difference between a signal and the Wasserstein distance between its diffusions. And we can extract these curvature properties, Olivier-Ricci curvature, from the heat dynamics or the wave dynamics. But I will tell you, we're still looking for a really good application of the chaotic dynamic, and we feel like it might be useful for modeling something more complex, maybe something to do with fluids. So if you have any ideas, let us know. Okay, so just to get you back a little bit to the actual application we're looking at, it turns out that a lot of the protein properties that I initially started with has something to do with these topological and geometric properties. So it turns out that you can also use the same kind of network to directly predict some properties of the protein, because there is some geometric or topological information that's important for the protein, but we don't know what it is. So if you bake those in, and if you're like, okay, I'm just going to measure curvature and then I'm going to predict proteins from it, it might not be as good. But because these dynamics already have contained information about a lot of geometry and topology, they might do well. So basically, I'm going to end my talk there. But I'm actually just going to advertise this paper a little bit. All the while I was talking about constructing a fixed graph, pretending you know the graph, but it actually turns out that we can also learn the graph. And while I'm at it, I'm going to advertise a little bit this conference called Learning on Graphs, where this appears. This is happening again. Many of you, I encourage many of you to get involved in it or submit. This year I'm on the PC. So the idea is we use a form of graph attention and dynamics prediction to relearn this graph. So with that, I'm going to end and go with questions. I lost my presentation just now, but if you have questions, I'll bring it back up. Thank you.
Any questions? Thanks, Peter, for this engaging presentation. You mentioned briefly about the inverse of the spectrum. How important is that for